And it is with my great pleasure to welcome Dr. Dwight Kravitz, a professor of cognitive neuroscience at George Washington University today. And Dwight's research expands a multitude of cognitive neuroscience domains, including visual object recognition, word recognition, concepts and scenes, mental imagery, and visual working memory. I'm personally a fan of one of his papers published recently in the connection of perception and visual working memory. And like you, I am excited to hear his talk today on predicting functional organization and its effects on behavior. With that, I welcome Dwight on the stage to start, and also remind you that you can use the Q&A panel to ask questions, and I will ask them at the end of the session. Okay, thank you.
Well, I want to thank everybody for coming, and thanks to Brad and Yalda for inviting me and pulling all this together. I know it's an incredible amount of work, particularly in the midst of a pandemic. So to jump right into it, this talk is really sort of a whirlwind tour of a bunch of different studies that I've done over the last 10 or so years that we're looking at specifically bridging the divide between cognitive science, the science of behavior, right, and neuroscience, which was and still is producing a number of fundamental insights about how the neural substrate behaves, how it can support complex behavior. But between the two, there's a vast gulf of complexity, right? We really do not have a firm handle on complex circuit behavior, and the entirety of systems neuroscience, certainly from a cognitive perspective, ultimately relies on trying to find aspects of the physiology which are correlated with behavioral performance, and then in theory also related to our sort of putative cognitive mechanisms. But in fact, the problem is really very difficult. The river itself, right, is constantly changing. It changes from person to person. There's no reason to expect that the solution of a neural network to a problem in one person will necessarily be the same in another. And as we'll see towards the end of the talk, it changes from trial to trial, where the relationship between the physiology and the adaptiveness of individuals' behavior is going to change in reference to the particular task context. And this, certainly in my career, has been a limiting factor in how tightly we can actually bridge this divide. The typical approach, I think, is we have some sort of putative mechanism, right? A theory that produces a hypothesis that we test in some particular instantiation. And then we go ahead and look and see, while the subjects are performing this task, what the activation of their brain is like, right? At least within the context of human neuro. And then I think oftentimes the temptation is to look at the pattern of activation we see and start ascribing particular functions, right, related to our putative models to the activation that we see. So we point at a particular blob of activity and say, aha, this is, working memory, or perhaps the central executive or the sketch pad or whatever it is. I think in practice, this approach, as the field has started to really acknowledge, has its limitations. What we're doing there is looking for bits of the variance in the physiological response that agree with what we thought there should be. And when we find agreement of any sort, really, we're happy enough to go ahead and report it. This is a good sort of first order approach, but I'm going to advocate for doing something a little bit different. So the approach that I've taken, along with a number of others, I think in this field, has been to try to take our physiological observations and then look in detail at the things that we know, right? So let's take, for example, the case of localization. So we have some area of the brain that's active in a particular task, but now rather than leaving it at that, let's instead dive into the known physiological properties of that region of the brain, see whether or not any of those properties then make some kind of a behavioral prediction, right? So rather than just noting that there's some variance that seems to agree with us, see whether or not the consequences of the engagement of a system are reflected in actual ongoing behavior. And ideally, that should be a prediction that's novel, something that you couldn't have made without the physiology. And that helps to bridge this divide, right? It helps to shoot the arrow from the other side of the chasm, right? Having managed to find its mark. And then eventually to work our way back to our original models and update them, right? Not just to sort of wholesale reject or accept them on the basis of the data, but to try to bring them into closer alignment with the known properties of the physiology. So I'm going to take you through a set of studies across 3 different domains where we attempted to do this, looking again at localization and category selectivity, the relationship between working memory and perception, and then finally at a set of domain general mechanisms and how those are going to play out in the context of attention and learning. So to begin with category selectivity, it's helpful to kind of set the stage. Going back to the late 70s or early 80s, our understanding of the primate visual system has been a bifurcation, with a dorsal going pathway responsible for spatial processing and a ventral pathway that's responsible for, say, recognition or identification. And the core data that established this dissociation, right, is the inability of monkeys with lesions in the dorsal pathway to learn tasks based on spatial adjacency, but they are unimpaired at learning tasks on the basis of recognition, and vice versa is true with ventral temporal lesions. This basic idea, right, of certainly the ventral pathway as a staged hierarchy, where what you're doing is proceeding from early visual cortex down the length of the temporal lobe through a series of processing stages, each one of which is getting more computationally complex and building more and more complex representation from the aggregation of the simpler representations rather, that are feeding into those areas, has proved to be very powerful, right? It's a relatively simple idea. It's elegant. There's a fair amount of data that sort of directly supports this characterization, where you have an increase in, for example, the latency of the first spikes, and an increase in receptive field sizes and the complexity of representations along the hierarchy. When we take this basic idea, which is really a sort of neuroanatomical extension of Hubel and Wiesel's ideas, and then we instantiate those into computational models, we get really powerful results. Models are quite capable at this point of doing really complex object recognition, even amongst complex scenes. That story, right, is therefore quite compelling. And it's largely an inductive story coming out of neuroscience, with some observations coming from, right, monkey neuroanatomy and monkey lesion work. But we run into a problem when we then look at the human data. So human fMRI, particularly sort of the early studies looking at category selectivity, were coming from the other direction, where they were looking for neural correlates of the sort of specialized processing for particular categories that have been observed behaviorally. And what they find is actually quite astonishing, right? It's a set of regions which are in stereotypical locations across participants, right? And each one of these regions appears to be selective or more strongly responsive for one category than for many, if not all, of the others tested. And the fact that these regions are also showing up in stereotypical locations, right, suggests an importance to this organization, right? It's not a totally arbitrary organization. But there's no way to go from the sort of staged hierarchy overarching model, right, of the visual system to clustered selectivity that's always occurring in the same location. And as a result, some proposals have emerged where this organization is proposed to be actually arbitrary relative to the neuroanatomy, which is to say, here in a paper from Edward Haan and Alan Cowey, right, they conceptualize the extrastriate cortex as a series of specifically evolved modules, each one of which comes into being in order to support processing of a particular type, and then is densely connected to other modules which share that sort of processing, but only loosely connected to ones which don't. This is a conceptualization that certainly fits with the fMRI data, but it produces a problem, right? The first is it has no underlying relationship to the non-human primate literature at all, you know, beyond just now running the same studies in primates and seeing if they also have these areas. And the other problem is that it opens up a sort of infinite quest for new regions, right? We can go out and now try to tessellate the entirety of the inferior temporal cortex with sort of object selective modules, but it's not entirely clear, right, that we're going to get any of a deeper sort of theoretical understanding of why those modules are there, right? In fact, this conceptualization sort of cuts that discussion off. So I'm going to argue actually that implicit in the data that already exists is the answer to this conundrum. The first hint is the WWFA, which is this really, again, very surprising region that appears to be selective for visual word form, right, over consonant strings or pseudo words or digits. It's also in a consistent location across individuals. There is a associated disorder that results from lesions in and around this area of the brain. But unlike the other areas, which are all sort of biologically relevant categories, right, this cannot possibly be the result of evolutionary pressure. Literacy, right, in the general populace in the United States is, charitably really 200 years old. And so there's not really been time for some sort of a mutation event that's going to enable the recognition of orthography. And so in order to resolve this and a number of other related conundrums, I undertook with a bunch of collaborators who you see down here at the bottom. And throughout the talk, there will be pictures of my collaborators without whom absolutely this work is impossible. Here in particular, I have to call out Salim Khattar-Bacha on the far left, who is simply encyclopedic of his knowledge of macaque neuroanatomy. And so with his help, we undertook to look in greater detail at the neuroanatomy of both the ventral and the dorsal pathways relative to the last 30 years of neuroanatomical tracer work that had been done since these pathways were originally characterized. And as we'll see in the details of this neuroanatomy, we begin to see hints of why this stereotypical functional organization emerges. So here is a sort of summary plot of that neuroanatomy. Don't try to take it all in. It's of course hideously complicated. In addition to the complexity that's internal to, in this case, the ventral visual pathway, there are also a complex set of relationships and projections in and out of it coming from external systems. Like the medial temporal lobe, the orbitofrontal, the basal ganglia, etc. And within this specific set of connectivity, we begin to see a reason why we might observe stereotypical structure even amongst high-level visual representations. And so in order to make this link, the first thing that we have to do is to recognize that the models that we're building, right, are models of a computational principle, right, an algorithmic approach to the problem of vision. What we're testing when we look at our deep belief or DNN models is whether or not a sort of staged hierarchy with these placed nonlinearities is capable of solving some of the problems of vision. But this is not really a model of the actual detailed neuroanatomy. And as a result, those models are not going to naturally produce the same functional organization that we see in humans. So the argument that I'm going to make here in this section is that natural selection, right, the evolutionary events occur at the level of the gross connectivity of the brain, right? They are in response to the problem of packing this many neurons into a person's head, which necessitates that only some connectivity is going to be possible. And that in turn is going to mean that information is only available, right, in certain parts of the brain about certain aspects of the visual world. Right. As a result, when you go ahead and now begin to interact with faces and scenes and body parts and so forth, there are certain parts of the extrastriate cortex that have better access to the information you need in order to do that sort of processing. And as a result, you end up with an experience-driven development of category selectivity, right, which nonetheless will still have a stereotypical location defined by the gross neuroanatomy. And so we set out to look at this experimentally, and moreover, to look at it in a domain where we could really take a data-driven approach that would allow us to see whether the predictions from the neuroanatomy really hold true without necessarily having to have a biased paradigm design, right, where it can only show us that we're right or not. And so we chose the domain of scenes. Scenes are great because they are exceedingly complex. Right, when you look at this, this room, for example, you have undoubtedly across the entire cortex responses to a bunch of different aspects of this scene, right? So you could be responding to the objects, to the category of scene, this is a hotel room, to the geometry of that scene, right? How navigable is it? How do I get from one side of it to the other? You could be grappling with the complexity that this is an enclosed space that has windows to the outside world and that you're a fair deal off the ground. right? In addition to being able to handle all of those different forms of complexity, there's also a huge amount of heterogeneity, right? Different scenes look very different from each other, and yet the underlying physiological response has to be capable of capturing, right, aspects of all these different aspects of the scenes and across very different scenes as well. So what we decided to do was look at part of the ventral visual pathway that is known to be highly scene selective, the parahippocampal place area. You can see its location here in a schematic, and then also in raw 770 EPI slices, right? This sort of blue swath here. And what we're going to try to do is say, well, we know that PPA is responsive to scenes, right, selective for them, but what we can do is now try to predict exactly what aspect of scenes PPA is going to be picking up on the basis of its connectivity, or the connectivity of the homologous area in the macaque. Right, so if we go ahead and now look at the macaque, our homologous area is, not surprisingly, the parahippocampal gyrus, which in macaque includes these two areas, TFO and TFTH. You can see here that they get the majority of their input from V4V. Not shown is that in detail, they're getting the majority of their input from the peripheral portions of the V4 ventral map. Okay, so that gives us some clue that they're going to be, these areas are primarily getting their information about the peripheral portions of space. We can also see some evidence of that in the human data, looking at these sort of classic studies from Rafi Malik's lab, where he shows that there is a center versus periphery bias with the medial aspects of the temporal lobe representing the periphery. We can also look at the descending connectivity coming into this area from the dorsal stream. It turns out that there is an evolutionarily ancient pathway, highly sort of studied in rodent, but also present in primate, that goes from the caudal portions of the inferior parietal lobule down through the retrosplenial posterior cingulate into TFO, TFTH. And you can also see the dense interconnections between those areas and the hippocampus. Okay, again, there is some evidence for this connectivity in the human, looking at functional connectivity. Here, a schematic from a study by Margulis and colleagues, showing in blue a large amount of functional connectivity between the IPL and the medial temporal lobe. And so in our study, what we're going to do then is predict that PPA should be really responsive to the spatial aspects of scenes. It's receiving sort of descending information coming from the parietal, deeply integrated with the navigation system, right? And now what we're going to do is present scenes, but in a way that is unbiased, right? So what we're going to do is present as many scenes as we could. At the time, that was 96. And we draw them from a wide variety of different categories, some of them enclosed spaces, some of them open. And what we're trying to do here is to just see what aspect of the scenes the region ends up representing. What we can do is use multivariate pattern analysis on each individual scene now, look and see how similarly the region's response is treating every pair of scenes, visualize that in this configuration where adjacency is reflective of similarity. Some of you can already see what this structure actually is. It's a little easier if I turn it into symbolic. So here's the same structure now with the scenes colored by whether they're man-made or natural, near or far, and closed versus open. And so very clearly there's a distinction here, right, that the region is holding between these enclosed versus open scenes, right, consistent with its kind of connectivity. What we can do then is now make the pivot I was talking about earlier and try to make a behavioral prediction. So if in fact this region is responsible for representing this distinction, then we should see its representation reflected in participants' behavior. And so what we did was to take a separate group of participants and arrange a series of matches where they were presented with two scenes and simply had to judge which one of them was more open, right? And then using the same math that they used to rank chess experts, what we can do is derive a linear ranking of how open or closed a scene is on the basis of this separate group of participants' responses. We can derive a similar metric, right, from the neuroimaging data where we see sort of how closely correlated the scene is with other open or closed scenes. And then we can directly correlate the two, and the relationship is quite strong. It's always good to have a control region. So here's early visual cortex, which doesn't show the same distinction and shows no relationship to the behavior either. Okay. So in this, what we've done is a data-driven approach to understanding scene representation, and we've shown that it's consistent with its neuroanatomy, but there are other aspects of this organization to category selectivity that we can explain. And so here's a study where, actually a set of studies where we were looking in particular at why these regions tend to come in pairs, right? At first glance, this seems highly redundant and not terribly efficient. The sort of existing accounts of this, the data has always been a little bit equivocal, but the idea is that perhaps the lateral regions are lower level than the ventral regions and are somehow accomplishing, say, an earlier stage of processing that then feeds into the ventral regions that have a higher order kind. If we look at the neuroanatomy, though, from the macaque, we see a different potential reason. So if we look at the lateral surface, we can see that it derives the majority of its input from the dorsal portions of the visual field maps. That is, of course, the lower visual field. The ventral surface derives the majority of its input from the ventral portions or the upper visual field. And so another reason why you might have these apparently redundant regions is because they have been shaped by experience to deal with these categories of objects when they occur in different locations on the retina, right? Namely the upper and lower contralateral visual fields. We can test that directly simply by localizing the regions and then running population receptive field mapping. And we see that this is in fact a difference which holds. What we see is that in the PPA on the ventral surface, there is a bias to the upper visual field, and TOS a bias to the lower visual field. This sort of argues against as well the hierarchical interpretation in that it's difficult for regions to be hierarchically organized if they're responding to different portions of the visual world, at least outside of fovea. All right, so that's my whirlwind tour of category selectivity. I want to sort of try to play the same game now, but in the domain of working memory. So the classic accounts of working memory very much hold working memory to be a sort of specialized system which exists distinct from the circuitry that's responsible for perceptual processing, right? So you have an executive, it has control of these slave systems that deal with specific kinds of information, but it's a sort of distinct process. And the neural models reflect that distinction. So they propose that initial perceptual processing is occurring in these perceptual circuits, but after that, information is going to be recoded and transferred into a frontoparietal circuit, which is specialized for maintaining state, right? We set out to kind of investigate an alternative possibility suggested by the sensorimotor recruitment framework, which is sort of actually a generalization of that, which is the idea that actually you maintain information not solely within the frontoparietal circuit, but within whatever circuit is most appropriate for holding that form of information. And so we take a very simple paradigm here adapted from the paradigm used by Frank Tong rather. And what we have here, right, is two samples that come from two different categories of visual object, followed by a cue, which tells the subject which one they should be holding on to during this delay period. Okay, and in both of the two different types of runs of this task, this sequence is exactly the same, right? What differs is simply the task demand of whether following the delay, participants have to identify whether a fragment of an object was part of the item they were holding in memory, or whether an object is from the same particular category, right, as the object that they are holding in memory. And so in the visual task, right, you are required to maintain visual detail, whereas in the non-visual task, you're required only to maintain some abstract category label. And our prediction is that we should see, using again multivariate methods, information during the delay within perceptual cortices during the visual task and within the prefrontal cortices during the more verbal task. Despite the fact that the lead up, right, to the delay and the delay itself is identical. And this is exactly what we find. So here in a object selective cortex, we see significant classification performance of the item held in memory during the visual but not visual runs, and then the nice crossover interaction with the opposite being true in the prefrontal cortex. Right, so this is good evidence that it's not the case that there is some specific system, right, that has been adapted in order to maintain state. In fact, any system is capable of, at the very least, reflecting, right, maintained information. However, what we would like to see, right, is a cleaner behavioral correlate of that as well, right? Is it the case that this information is really indicative, right? The presence of this information is really indicative of that circuit's involvement. And so let's consider then the behavioral predictions that emerge from that result. If in fact working memory and perceptual processing are occurring within the same circuits, then we expect there to be bidirectional interference between the two of them, right? What you are holding in mind should alter what you see, right? And what you are currently seeing should alter what you're holding in mind. And moreover, the amount of interference should be commensurate with the overlap in the neuronal populations that are engaged. And since we know a fair amount about the tuning of these areas for at least low-level phenomenon, we can predict, right, that there should be more interference the more sort of shared cells, and therefore more similar the stimuli are, at least according to these tuning curves. So I'm going to show you that prediction holds across 3 separate experiments. So the first thing I'm going to show you is that ongoing perception is going to bias visual working memory in a way that actually directly recapitulates these tuning curves. And so then it's a very simple experiment. Participants are presented with a memory cue. They have to hold on to it. And then during this delay period, before they have to report the item they're holding in memory, they're going to do an RSVP task where a series of letters is going to appear. And whenever one of those letters appears in white, they have to press a button. Behind this RSVP stream, and totally irrelevant to the task, is another colored disk, right? A distractor. And what we're going to do is systematically manipulate the similarity of that distractor to the memory cue with the anticipation that we should introduce a greater distortion or bias, right, in the working memory report, the more similar the distracting cue is. Okay, and then following this, they're going to do this continuous memory report. Right, what I'm going to plot for you now is the amount of bias that's introduced by the cue as a function of how dissimilar it is. And we get, of course, exactly what we expect, which is this nice monotonic decrease with similarity. This is this game played out in color. We can play out the exact same game in orientation. And again, we see pretty much the exact same result. Okay, this shows us that, in fact, the tuning curves are reflected in this interference between perception and working memory. Now I want to show you the other direction, that working memory is going to alter ongoing perception. Okay, so in order to do that, we're going to test for a distortion introduced into perceptual discrimination thresholds by holding an item in memory. And so I apologize in advance, it's rather involved, but we'll get through it. So participants will be shown a colored Gabor, right? And that Gabor has both an orientation and a color. They are tasked with holding in mind one aspect of it, either the color or the orientation. Following a delay, they will then be asked to do a color or orientation discrimination, where again, they are presented with 2 colored Gabors, and they have to say whether they are the same or different on the basis of one of those features. Following that, they do a continuous report, again, for either color or orientation, depending on what their actual task was. What we're going to do is systematically manipulate the similarity between the discrimination stimuli and try to derive the discrimination thresholds as sort of an extremely low-level perceptual task. And then we're going to see whether or not we observe a predicted effect, right, of what you're holding in memory on those discrimination thresholds. Moreover, as is my habit, I look for crossover interactions and I design for them. And so what we're also going to do is now systematically manipulate whether the item you're holding in memory, the feature you're holding in memory, is going to match the feature upon which you're doing the discrimination or not, right? And so if you're holding color in memory and doing color discrimination, that's match. Orientation is match, but you can also have these non-match conditions. And in the non-match conditions, we predict that there shouldn't be, right, nearly as strong an effect. In fact, it should be largely absent because you're not actively engaging the same population of cells, which you're then going to use to do the perceptual task. Okay, and so how are we going to test for this effect? Well, we're going to take advantage of our previous result, which shows that the amount of interference is going to be proportional to the similarity between the items held in memory and the items you're currently perceiving. And so we're going to have 2 conditions. In the middle condition, right, in both conditions, we will present the same discrimination stimuli represented by these sticks here on this color spectrum, right? In the middle condition, the item you're holding in memory is going to be between the 2. And what that's going to do, right, is draw the representation, right, of the 2 discrimination stimuli towards itself. In so doing, the actual distance is going to actually be larger than the perceived distance. Then we can contrast that when to the case where you're holding an item in memory that's off to one side of the discrimination stimuli, in which case there should be a greater shift, right, for one of these stimuli, one of the discrimination stimuli than the other, and thus the perceived distance will actually be greater. And so in this case, we predict higher discrimination thresholds because it's harder to distinguish the stimuli, and here lower discrimination thresholds. And I'll accelerate a little bit and tell you that that is exactly what we find. So here are the raw psychophysics curves for the middle and side conditions, middle in blue, side in green. You can see that side has a significantly lower threshold, right, than does middle. This is for color memory. When we do the non-match condition, there's no difference between them. Okay, when we do now we look for orientation memory. Again, we see the difference when there's a match condition, and the difference is absent when you're in the non-match condition. We can plot that as the crossover interaction, and you can see very clearly what this effect is, right? So ongoing perception is going to affect working memory, introducing a bias, and it is also going to be affected by working memory, causing slight distortions to be introduced. And this is all, of course, commensurate with the co-localization of these processes, right, to the same areas. I'm also going to show you that these effects persist across delays. So in the recoding hypothesis, over time, you're moving information out of these perceptual circuits. And so over time, there should be weaker effects, right? Whereas with sensorimotor recruitment, right, you're using the same circuit. And so over time, the only thing that's going to happen is the working memory representation is going to decay, right? But you'll still get these interference effects throughout. So for the threshold effects, right, we're going to expect both of these models to predict that the effect should reduce over time, though recoding does predict that they will eventually extinguish totally. But the bias should actually increase according to the sensorimotor recruitment hypothesis because you have a decay of this working memory representation. Whereas the recoding approach would predict that the bias should also be extinguishing over time. We can test this very simply by adding a variable ISI to the experiment that we've already run between the orientation and the memory and discrimination task. And we get exactly what we would predict from the sensorimotor recruitment story. So over time, the color discrimination effect reduces while the bias effect increases. This is a summary plot of those two things on the same axis. And so you can see that even though there's a reduction in the threshold effect, it stabilizes and it is always significantly above 0. We have run versions of this task out to 6, 8, 10 seconds, and the result holds. Okay, so in total then, what we've done is taken a co-localization that fell out of fMRI data, and we've used it to make a novel behavioral prediction, right, that holds up across a number of different experiments. And it reveals what I think is a really novel and profound interaction, right, between these two processes that we would not have been able to discover were it not for the physiological data, right? And then I think just very quickly, no, there's really not going to be enough time to do justice. So I will leave this last section, and I will only sort of précis it by saying there is a paper which is deposited on the SciArvix, for which there is a link in the chat window that describes this work. In summary, what it is about is showing that the low-level consequences, right, of synaptic response, where what you have is necessarily as a population of cells is firing a longer term, well, sort of a short-term potentiation of those synapses due to calcium kinetics, but then also a long-term sort of Hebbian learning, which is obliged to always occur. As a result of those, what you can see when you look at big data, right, and you have enough data to look for these subtle effects, is that over time, subjects are about every single feature of the display, actually, ones that are both task relevant and task irrelevant, they are accumulating evidence, right, for the presence of a certain feature. And their behavior on a current trial is going to be profoundly influenced by the history of trials that came before, in a way that is actually strikingly consistent with basic metrics of statistical evidence, and the time course of which is also consistent with known mechanisms of synaptic plasticity. But beyond that high-level summary, there's not really enough time, but we can go into it during the discussion section if we want to. I'll skip, oops, sorry about that. And so what I want to leave with is really just this sort of summary statement. what we've got now is a huge amount of data and a lot of really careful experimentation across the entire field, where what we're looking for are correlates of our sort of putative behavioral mechanisms. Where we've run into limitations has been really with the sheer complexity, right, of the physiological data that we're able to collect. The data is itself a very low dimensional cut through very complicated brain response. It's colored by the particular paradigms that we use. And it is also malleable, right? It changes from moment to moment, as does the behavior of the subjects in our experiment. And so my approach has been, and I think I'm not unique in this, to search for those aspects of the physiology that make specific predictions, right? That are going to hold true even over all of that variability across individual trials and across individual people. But I think that we will need to increasingly, as we did in this final project that I ended up having to skip over, I think we will increasingly have to grapple with that variability directly. We will need to understand what is actually changing from trial to trial and model it specifically to understand what aspects of the physiology are related to stable cognitive mechanism versus what aspects of that response are really capturing more implicit learning, right? That's automatically happening at a local circuit level. And in general, what we need to do is test these really specific behavioral predictions that fall out of our physiological work in order to really bridge this divide much more effectively. And so with that, I want to thank everyone for their attention, and I'm happy to take questions. And if I don't get to a question, just shoot me an email. Thanks.
Thank you so much, Dwight, for the interesting talk. Now we can open the questions. There is one question on Q&A that I see Hazel Jackson asked. So the question is, I may have like basically missed
so
like the macaques or human studies.
Sorry. So yes, I see it. The macaque work is really all neuroanatomy. The rest of the empirical work is all done in humans. So the work that comes from macaque is really an aggregation of 30 to 40 years of neuroanatomical tracer work, but all of the neuroimaging and the behavior, that's all humans.
Mm-hmm. Okay, thank you. So if you have, like, I'm talking to our audience, if you have any questions, please feel free to send them on Q&A. I will read them. Meanwhile, I have a question, Dwight. You had some very interesting results on organization of like category regions and its connection with maybe even topology of visual fields, right? If I got it correctly. So I want to know more your insight on that. Like, for example, at one point you said TOS has a more higher visual field bias compared to PPA, which has like lower visual field bias. That's a very interesting finding. Can you comment on more on that?
Sure. So one way of thinking about this in general, right, is that we want to have an inductive account, right, that stems from the known properties, right, and the better understood properties of low-level visual cortex, and nonetheless makes a prediction, right, about the functional organization that we're going to observe as we move sort of higher up in this visual hierarchy. And we kind of began this actually looking at retinotopy and how sort of position invariant object recognition actually was. And in fact, we found that there was not very strong evidence for that position invariance. Certainly, particularly when you looked at tasks that were more visual versus semantic. And that led us to this sort of long project where we were looking at the potential relationship of retinotopic maps, right, to category selectivity. And when you think about the categories, they come with a bunch of sort of stereotypical retinotopic locations, right? So when you're looking at, for example, bodies, right? If you're looking at a person, you typically fixate their face, which places their body in the lower visual field. And then you look at, say, body selectivity, and the lateral body area is much more reliable and robust, representing the lower visual field than the ventral body area. Likewise, right, the left side of the body ends up in one visual field and the right side in the other. We ran another multivariate study looking at information for body parts and found there was greater information when they were in their stereotypical low locations. And so to my mind, I think that the sort of category organization that we see in the extrastriate is reflective, right, of these lower level constraints, right? In essence, the banks of the river. So even though the river changes from moment to moment, and it probably changes certainly at a micro level from individual to individual, at a broad scale, gross connectivity defines the banks, right? There is only certain areas which are going to have access to the right kinds of information. Combine that with experience and frankly, a task demand to deal with this stimuli in the real world, and you end up building clusters, right, which are going to be all processing similar things because they have access to similar kinds of information, and they've been shaped by experience to do that. And so, you know, pretty much every time we've gone looking, be it in behavior, fMRI, neuroanatomy, we see this sort of a linkage. And in fact, as we got more involved in developing imaging methods, right, as the data became cleaner and we got better views of this, of the ventral surface in particular from our 7T data, the more this appeared to sort of hold up, right? The less and less they look like isolated islands sort of arbitrarily placed, and the more they look like processing streams, where what you've got is sort of a peripheral processing stream that is somewhat specialized for scenes, because that's primarily what you do in your periphery, is process these spatial aspects of scenes, right? And then sort of foveal stream that's devoted to words in one hemisphere and faces in the other.
Okay, thank you. We have a couple of more questions in Q&A. Can this bias considered as a confabulation mechanism related to poor memory, or is it all due to perceptual mechanisms?
So, I mean, I think if you're talking about it at a pure behavioral level, right? If you just look at the behavioral paradigm, then the answer to that question is ambiguous, right? It's difficult to say whether or not you have some sort of a distinct memory representation, right, which is degrading and it's poor and it's being impinged upon by ongoing perception, or whether what you have is the sort of, as proposed, this mutual engagement of the perceptual circuitry. I think ultimately the argument rests on a combination of the behavior and the imaging result. So I think the imaging result says, you know, look, we are equally capable because of this crossover interaction of detecting information in either of these systems, but we really only detect it in this perceptual area during the perceptual task. Then we make this set of behavioral predictions that falls out of the sort of tuning curve properties of those perceptual areas. And so the sort of simplest account, I think, is the one which I gave. If you sort of want to maintain the notion of a distinct system, then I think you have to grapple with, you know, not only why is it that ongoing perception is impinging on that system at all, but then also why is it that the adjacency rules that come from the perceptual tuning are also in this system. And so I think this is one of those cases where, you know, as, you know, not one of those cases, as is always the case, there's no silver bullet result, right? And I wouldn't want to just take the behavior and make that argument. But I think in combination with a bunch of imaging work now, I'm more comfortable with that.
Okay, thank you. So the next question is by Hajar, and the question is, in the world of big data, it will be harder to conduct very controlled experiments. How do you think we could incorporate your arguments during this talk with population-based neuroimaging data?
So I think, all right, there's two parts to the question. I'll do the second part first. So in terms of using population-based neuroimaging data, you know, that's actually what effectively, too far. Sorry, there we go. That's effectively what this prediction is, right? What we have here is across a population of subjects in fMRI. This is the average response of the PPA, right? This is on average how similar, how open or closed it treats any individual scene from amongst the 96. And then in a separate population of people, right, again, in a controlled laboratory experiment, what we can do is test and see whether or not the specific representation we derived from the population fMRI holds in the behavior. And so I think there are opportunities, right, to do that sort of work where what you're looking for are going to be aspects of the population level physiological response that do make these particular behavioral predictions. Now, for the former part of the question, you're absolutely right that in the world of big data, you're not going to have laboratory levels of control. That's not going to happen. I think, though, that when we think about big data, we need to think of it as a complementary kind of mechanism by which we can study this. So the advantages are the sheer volume of data, which allows you to overcome some of the issues of a lack of control with the sheer power, right, of the sample that you can collect. So to give sort of an example of that and sneak in a little bit of detail on this final paradigm that I didn't get a chance to get to. Too many animations. There we go. So what we're doing here, right, in this plot is we can look at now behavior on every trial, because we have in this plot 50,000 people, in some other plots, right, 1 million people who have done trial 1, or 15 million people who have done trial 1. And so in the context of big data, I think there are opportunities to look at trial by trial effects, to look at in general effects with a level of fidelity that we've not been able to do before. Where we run into the issues, right, are we want to then see, right, from the big data, a set of predictions that do then hold in our more controlled laboratory experiments as well, right? What we don't want is to have big data sort of spin off into its own subdomain and then have the two camps begin to ignore each other on the basis of either a lack of fidelity or a lack of control, right? Ideally, what we're looking for is going to be results that fall out of the big data that then lead to predictions in laboratory experiments and vice versa. And this project, of which again there is a preprint deposited on the archive, is a start at doing exactly that.
Okay, thank you so much. I think we are like done with the time, like 4 minutes. At the end, I would like to again to thank you, Dr. Dwight Kravitz, for joining us today and very interesting talk. And thanks the audience for the great questions and attending this session. So that's it. Bye, everyone. Have a great day.
Thanks, everybody, for coming.